Hi guys, welcome to Methods of Studying Cells. So as you can see, the specification is quite heavy. There are many skills that you need to be aware of. So like microscopes, eyepiece reticule, the drawings, the ultracentrifugation. So let's get started. Okay, so we'll start with the picture. I've got the picture here and drawing, and you need to suggest two ways in which you could improve this drawing. Okay, so that's a really common question. So don't use shadings, use only single lines, don't use sketching, add further labels, annotations, don't cross label lines, and add magnification. So that's a really good question to get a few easy marks. So, what is cell fractionation? It's the process of separating organelles, and this is obviously easy topic that you all probably know, but you're making mistakes for always saying the word cell. When we want to use organelles, like name them, so like mitochondria, like nucleus, and so on. So in terms of the process, it's divided into three main stages: homogenization, so breaking up the cell; filtration, getting rid of big bits, so here we mean large debris; and ultracentrifugation, separating the organelles, so here when you need to name them. Okay, this is how the process will look like. Okay, so starting with step one, and what's really important here, we'll be looking at cold buffered solutions. Okay, so there is a reason behind this, and obviously we will be looking at the speed. So when you speed at the first time, you do it at low speed, so the heaviest organelles will go to the bottom of your tube that you're going to use here. That will make appearance. So for example, nucleus, then you're going to speed at the higher speed, okay, to separate your less dense organelles. So as you can see here, there are different speeds. So the nucleus will be separated at the lowest speed. Then we've got mitochondria. Then, for example, lysosomes. So nucleus, it's the biggest. Mitochondria, it's slightly smaller, but lysosomes are much smaller than the other two. That explains the speed that we need to use. So homogenization then, it's the process what we say we need to use the ice cold buffered solution. Why? So ice cold to reduce the enzymes activity that break, and here it's that linking. Don't just say reduce enzymes activity, refer to the question. So prevent the breakdown of, for example, mitochondria. Isotonic, so the same concentration, the same water potential, okay, so prevents osmosis, so the named organelle, it's not going to be damaged, for example, not going to shrink or whatever. And buffer solution, so maintains the pH, so again you could refer this, so won't denature proteins or enzymes. Okay, and as the result of this, this breaks up the plasma membrane and releases the organelles into solution. So here we've got the model answer. Describe and explain how cell fractionation and ultracentrifugation can be used to isolate mitochondria. And look, be careful. This is the name organelle here. That's what we want to then use in our answers. Okay, so you can pause the video now and check if you can do it. Right, ready? So the answers: cell homogenization to break open cells, filter to remove large debris, and make sure you include the word large. Okay, use isotonic solution to prevent damage, look, to mitochondria. Keep cold to prevent reduce enzymes activity. Okay, so prevents the protein or enzymes denature within that mitochondria. Centrifuge at lower speed than nucleus. Sorry. So to separate nucleus for the lower speed. Okay, and respin at the higher ER. Look, ER. Okay, to separate the mitochondria, which will be found at the bottom in the pellet. So here again, why would you use those steps? So homogenize, break open cells, filtrate, remove large debris, cold reduce enzymes activity, isotonic prevents osmosis, so doesn't affect the water potential. Right. Again, same question, so they keep repeating themselves. So why would you use those ice cold buffered? Okay, same water potential, same model answer. Explain why biologists use the blender and then filtrate. So blender to break open cells, a filtrate to remove large debris. Name the organelle that will be in the first pellet, always nucleus, and the second, okay, centrifuge tube was spun at a higher speed to obtain the sample of organelles in labeled C, and suggest why, because it's less dense, okay, than nucleus. So you need to compare it to where is it less dense. Map skills then, so magnification, I'm not going to go through explanations of how to do it because it's only one tip here that I need to give it to you. So conversion, remember 1 centimeter equals 10 millimeters equals 10,000 micrometers, and please remember that adding 1 0 and then adding 3 zeros, so that's the easy conversion. And then my only tip for magnification is whatever you measure with ruler divided by real, right? So ruler, whatever picture is given to you, measure with the ruler, convert the units to micrometers, and then you can work out, for example, the real size by dividing whatever you measure with the ruler by given magnification. That's the easiest way of doing any questions with magnification. Right, so microscopes now. So two types of the microscopes, TEM and SEM. So transmission electron microscope and scanning electron microscope. So we need to be aware of those, and we need to look at the limitations of those and differences. So first we'll compare the TEM with the optical microscope. So TEM uses electrons, optical uses light, has a higher resolution, but not here. Smaller organelles can be observed in great detail, not for the optical microscope. You can view only dead specimens. You can view live specimens for the optical. Doesn't show color. Optical will do. Requires thinner specimens, but it doesn't here. Requires more complex preparation, but that one doesn't, and focuses using magnets, focuses using optical glass lenses. So make sure you know it, because then examiner can ask you many aspects. So if you know this table, you can easily then apply your answers to many different questions. So next questions, we're looking at the limitations of TEM and SEM. So limitations. So obviously what we've said, you cannot look at the living material, and you don't do it for SEM as well. Specimen must be very thin, okay? For the limitations of SEM, obviously it doesn't have to be as thin as electrons do. So a specimen does not need to be very thin because electrons do not penetrate. So then what we've got, so image will be only in 2D, but we will have image in 3D for the SEM microscope. And what we've mentioned before, TEM requires a complex preparation, same applies to SEM. Okay, so make sure you know your limitations as well. So here we've got another question. Describe the principles and the limitations of using a transmission electron microscope to investigate the cell structure. Complete sentences. So you can pause the video now, and let's have a look if you can apply our keywords. Right, ready? Okay, so first, principles: electrons pass through thin specimen, denser parts absorb more electrons, denser parts appear darker than, electrons have short wavelengths, so give higher resolution, and limitations. same answer. So cannot view the living material, must be very thin. Artifacts present, complex staining method, so long preparation, and we can view it only at 2D image. So look, if you know your tables, you can answer any questions. Right. So here, advantage of using TEM rather